Yeah, so lab 2 is about resource acquisition. So you were introduced to this topic in your pre-labs, and you learned a little bit about the different ways that different organisms acquire essential resources. So we'll dive into that, but first I just wanted to get anybody's questions about lab 1. How did the lab go? Are there any questions about the sampling or the post-lab you didn't understand, or even stuff about pre-lab that you wanted to go over again? So just take a second to think about if you have any questions, and you can type them in the chat, or also just unmute yourself and speak. Not seeing anything. A few reminders. So those, the post lab is due tomorrow by the end of the day. And then I've gotten a few, and so far looking pretty good. Just remember to, if you need to look back at the YouTube videos about how to make a rarefaction plot, or your post-lab materials, or sorry, pre-lab materials, just kind of review those as you're doing them. And then just as a reminder from what I'm seeing, just on your pre-labs, when you're submitting, please put the correct lab section number on your pre-labs. Otherwise, I may not be able to find them, and they may be listed as missing. So just really remember that, especially for this section is B08. And so a few people I think have been putting A08 just because it's the first one that appears, but just remember if you're B section, put B08. And then type your post-lab answers in what color other than black, just so I can easily see it when I'm grading it. I'm pretty sure that's on the instructions there as well. And then definitely just remember to follow the lab instructions as closely as possible. Some things to keep note of is make sure that you've done the correct number of samples that it says, follow the instructions for making the graph, that kind of stuff. But it'll tell you everything that you're expected to do. Just make sure you're reading the instructions closely. All right, so any questions about the post lab? Any questions about lab 1 in general before we move on to lab 2? Okay, awesome. All right, so diving into resource acquisition. Oh, sorry. We had a question about where we can find the recording for lab 1. So the recording for lab 1 is going to be in the YouTube channel. I have made like a section YouTube channel, and I sent out a one of my first messages has a resources list that has the YouTube channel link on there. So if you look through your old messages, it should be there. Messages from me. If you can't find it, just send me another message and I will forward it to you. Okay, great. Yeah, that's a good question. So the recordings will all be up on there. Okay, so diving into resource acquisition, important things to know. So there are different ways that different organisms can get their nutrients, and we refer to them by different terms depending on how they do it. So can anybody tell me what a heterotroph is, or what we mean when we say heterotroph? You can call it out, you can say, type it in the chat. Yeah, no, great, thank you. Yeah, I'm seeing something that eats something else. Perfect, well said. Yeah, so basically a heterotroph is they get their resources by consuming other organisms. So it could be from other heterotrophs or autotrophs. But yeah, so we are heterotrophs. We eat plants and other animals or whatever we eat, and we're able to get nutrients from them. So heterotroph. Can someone give some other examples of heterotrophs besides people, which I just said? Cows. Yes. Very Davis answer. Go Aggies. Any other fun favorite animals that people have? You can type them in. Dogs. Love dogs. I'm getting a puppy next week, so hopefully not for the next lab section, but the lab section after, I can show my puppy to everyone as an incentive for you to all come to lab in 2 weeks. So someone's favorite animal is their succulent, but not an animal. Good. So actually, this kind of leads into the next category. So autotrophs. So would your succulent be an example of an autotroph? Yep. Yeah. And so an autotroph, as we were saying, is different from a heterotroph. So it doesn't get its nutrients from consuming other organisms. It gets them mostly from the sun, but then there are also chemoautotrophs. They can get them from other chemicals. But so yeah, when we're talking about autotrophs, we're mostly talking about plants, and we're talking about getting solar energy. So this is kind of leads into the next question of how does each heterotroph and autotroph, how do they obtain energy and nutrients, and what structures and processes do the cells of each type use to obtain energy? So they kind of talked about, in the pre-lab, it talked about photosynthesis and then also digestion as two different processes that heterotrophs or autotrophs will use to get energy from the things that they consume, be it solar energy or, little nuts like the mouse on this screen is eating. So definitely these are important things to review. These processes are important for you to kind of, have a good grasp of as you're going through the lab. Any questions about these terms or the processes or comments about these things? Okay. So this lab is, somewhat different from the last one. And this is probably going to be a shorter lab section just because it's a little more straightforward. This in person would be a series of stations in the lab, and it's really fun because you get to interact with different critters and plants and kind of see these things in action. But the next best thing in our remote setting is that there will be a series of virtual stations focused on a particular concept. And I know, no critters. I wish that we could have sent those in the lab packs, but we can't. So you'll have 2 videos, one for station A through F, and then one for station G through L. And there are, in the lab manual, I think it talks about like the different points in each video that covers each station, so you can go back and review as many times as you want. And then there are these kind of review questions in the video as you're going, but we aren't grading those. I guess there have been a few problems with submitting them, so you don't have to do them if you don't want to, but they can be good for you to make sure that you're understanding the concepts and reviewing them. So don't worry about it if for some reason the submission from those questions on Aggie Video are not going through. And then as in our first lab, your post lab will be uploaded to Canvas as a PDF or a Word document, no pages documents, please. And then just kind of as you're going through these, something that's important is to think about the big picture of each station. Like, you don't need to get too caught up in the minutia of the organism. So like specific parts of each organism or stuff, but it's kind of about how the organisms relate to the concepts we're being introduced, how each organism is acquiring resources, and what kind of the overarching theme of that station is. So BIS2B is about a lot of big picture stuff. So BIS2C is where it gets a lot more into like memorization and like really knowing the names of things. But this, we're trying to get everybody to understand concepts. So just a quick overview of the stations. So you'll have autotroph station, A through F. So talking about light as an energy source and how capturing that energy goes. Then for B, you'll be observing photosynthesis, learning a little bit more about that process. So kind of not directly, but can be considered equivalent to digestion. I always really wish that I could photosynthesize. Like, wouldn't it be great to just be able to go out and sunbathe and like feel full from that? But alas. For station C, you're looking at single-celled, colonial, single-celled, colonial, and multicellularity. So I think this is looking at like teeny tiny guys. For D, roots and nutrients uptake. So in plants, like how the roots can help contribute to the nutrient uptake. So plants don't just need sunlight, they can't get all of their nutrients from that. So they also need to take up minerals and stuff from the soil, and that's what roots are good for. Energy storage in plants. So can anybody think of a structure in a plant that is used for energy storage? You could think about things that you eat that come from plants. If you can't think of anything, that's okay, but just take a guess. You can do it as a private message to me if you're worried about it being wrong. So, yeah, I might have misunderstood. Yeah, so there are, so basically I'm thinking of like a vegetative part of the plant that they can use for storing energy. Not where they make energy, but where energy, like sugars, could be stored, for example. Could be some in the stems. So a good thing that I'm thinking of is like a potato, for example. Yeah, exactly, in the roots. Yeah, so like tubers, these are great ways for those plants to store energy, and they also end up being great for us to eat, and there's a lot of sugars stored in them. So yeah, you'll learn more about it in the lab. So that's totally fine if that was I probably worded that question weirdly also. Okay, so then also how plants respond to environmental stimuli. So this could be, for example, you might notice in your houseplants that they lean towards where the sun is shining, so they kind of grow towards that. Different plants in drier environments can grow longer roots so that they can reach the water. So these are kinds of examples of how plants respond to environmental stimuli. Okay, so then you'll have heterotrophs. So you'll talk about food capture and digestion, single-celled heterotrophs. There's more food capture in different kinds of animals, insects, arachnids, snails. Then you'll talk about things with teeth. And then there's one station about fungi, so decomposers and parasites. And then a little bit about gas exchange and O2 transport, which were also subjects that you talked about briefly in the pre-lab. And then kind of overall themes that we'll be talking about are the surface area to volume relationships. So by the end of the lab, you should be able to calculate the surface area to volume equation. And then what are different adaptations for obtaining energy, both in plants and animals. So just, and I mentioned this earlier, but just as a reminder, the videos and lab manual will walk you through these stations so you can learn more about each group and how they get energy. So everything will be basically off of the 2 videos that were linked earlier. And I believe that those are linked in the lab manual on Learning Tree as well. So that's where you'll be able to just click and go. And then as a reminder, you're not graded on the questions in the video. So those can be helpful, but don't worry about them. And then focus on the big ideas. Any questions about any of these stations? Okay, great. All right, and so that was all about lab 2. For lab 3, just kind of as a heads up, please be sure to have gone through the population growth lectures and be sure to read through the lab before we meet. So just to make sure that we're all on the same page, just, a quick once over read, but there's 2 parts to it. So you're going to practice with estimating population size, and you're also going to learn about how to develop a hypothesis. So reading through the lab will kind of get you in a good space for being able to state a hypothesis, and then we'll talk about testing a hypothesis and being exploratory scientists like we all can be. And then I know this equation is scary, but we'll go through it, so do not be afraid. Any questions about this? I mean, don't worry about the equation right now, just about doing lab 2, reading through lab 3, etc. Okay. All right, so that we'll be doing derivatives. I don't know if you'll actually have to calculate the derivatives. It might be more like you're given a growth rate, but you will, as you'll all know, that is a derivative rate of change, but I don't think you'll be expected to calculate the derivative itself. Yeah, I don't think that you have to have taken calculus to take this class, do you? No. Okay. Whew. It's been a while since I've taken calculus, so. All right, so just as here's some happy grass cells. Look at how happy they are. Ah, so cute. That's all the material. Yeah, this lab is unfortunately a little bit harder to adapt, but hopefully the videos will give you a good sense. It's pretty straightforward. Just kind of, take it in, learn, do the activities that they tell you to do to get all these ideas about resource acquisition. Remember to turn in your post labs. Remember to turn in your pre-labs for next lab by Monday. And then I've noticed that there's a few missing pre-labs and also the lecture homework. So just be sure that you're following the to-dos on Canvas, keeping track of what dates to turn everything in, because I know that the instructors are pretty strict about the deadlines. And if you have any other questions, let me know. All right, so that's all the material I have. Feel free to stay on for some questions, or if you want to chat about any of the topics we talked about. But otherwise, have a wonderful week, and I will see you next week.